Good morning, friends. Today we'll be talking about the concept of ABO and Lewis blood group system. Very basic system. All of us are well versed with ABO blood group system. I'll be touching briefly the genetics part of the ABO group system and also the Lewis system, how it works, how it functions, and how this ABO antigens are formed, and how Lewis is pertinent to this ABO group system, and why I'm taking Lewis along with the ABO group system. So you can see that the ABO system is the most important of all blood groups in the transfusion practice. It was discovered by Carl Steiner, who is the discovered the ABO group system in 1901. The ABO system consists of ABO phenotypes A, B, O, and AB. We all know that these are the four groups, blood groups that we always talk about. That is the ABO, A, B, O, and AB. So what are ABO antibodies? These are naturally acting antibodies. These are IgM in nature, and they activate complement. They activate complement at a very wide scale, and they react at room temperature or a cold temperature, because IgM antibodies react at 4 degree or colder temperature, and they are detected with strong direct agglutination reactions. Now, the proportion of ABO antibodies is initiated at birth, but titers are generally too low. So the titers keep rising with the age, and so they are not detected until 3 to 6 months of age, and it peaks in around 5 to 10 years of age. So much so that the extremes of the age, that is very young age, like infants and the elderly, have non-detectable antibodies. So group O individuals, we all know this since our high school. Group O individuals contain only anti-A and anti-B antibody, but they also have one weird antibody called anti-AB, which is different from anti-A and anti-B. So this is a combination, and this is primarily IgG antibody. So you have anti-A, anti-B, and anti-AB, which is IgG. So anti-AB is a separate cross-reacting antibody that is usually IgG in inheritance of ABO group system. It was from Bertsin in 1924. It described the locus of the ABO gene is present on chromosome 9, and O gene, which is an amorph, that means it does not produce any detectable antigens. A gene will produce A antigen, B gene will produce B antigen, but O gene is an amorph, so no antigen is detective. And group O phenotype is an autosomal recessive trait with the inheritance of two O genes. So I'll be explaining this more. So these are the genes that are inherited. You can see, all of you can see that this is, if you see, if you are a blood group phenotype A, you can either be homozygous AA or you can be AO. So both of them will be represented as A. If you are phenotypically B, then you can be either BB or you can be B with O, because O will not, it's an amorph. And similarly, you have an AB where both A and B are codominantly expressed, and that is how the antigen AB is formed. But if you see that O, you can see that you have to have O is an amorph, so both the homozygosity of both O will be required to actually form a O phenotype. Coming to the genetics of ABO system, it is comprising of two chains and three genes. So please remember there are two chains, and on these two chains there will be three genes that are acting to form what is called as an ABO group system, but also a closely associated system called H system, or the H antigen, or the H system. But we invariably make ABO and H together, and we consider them as one system. So there are two long chains. Just I'll explain them further. Two long chains that are type 1 and type 2. Now type 1 chain, it is present generally in secretions and plasma. How do you remember? Type 1 means single, 1 means single, and single means solo. So they are freely floating in the plasma or secretions. Whereas type 2 chains, you mean 2 means double. So double, you can see that they are linked to something. So they are generally linked to the RBCs. So the RBCs or the RBC antigens are formed on type 2 chains, whereas in secretions there is type 1 chain. So that is how we remember. So type 1 and type 2, there are two chains. Now what are the genes that act on it? So we all remember through one thing, fucosyl transferase is the gene. So first gene is fucosyl transferase 1, the second gene is fucosyl transferase 2, and the third gene is fucosyl transferase 3. Now we can also call it foot 1, foot 2, and foot 3, just to shorten the things. Now H gene is present on chromosome 19 and ABO gene is present on chromosome 9. but they act together, and how they act together to form an ABO system is what I'm going to explain. So always remember, H gene is the FUT1 gene or the fucosyl transferase 1 gene. The secretor gene, SE gene, is the FUT2 gene, and ABO gene is which is present on chromosome 9 is the third gene that is involved. So FUT1, FUT2, and the ABO gene, right? There is another gene called FUT3, which is important for the Lewis system. That I have a slide where I'll be explaining that. So let's forget that for now. So we'll talk about just three genes. They are FUT1, FUT2, and the ABO gene. FUT1 is H gene and FUT2 is secretor gene, right? So what is the central dogma for ABO system? Always remember, this is a peculiar blood group system. What happens in this is there is a gene. Now gene here codes for an enzyme. This enzyme will act and produce a sugar on the existing whatever chain you have. So what this enzyme will do is it will add a sugar to it and thereby cause some change in that existing precursor substance and lead to a formation of a different antigen. So there is a gene which produces an enzyme will produce a sugar, add a sugar and make a separate antigen. This is the basic crux of this ABO system. So that is why we need to know what are the chains and we need to know what are the genes involved, FUT1, FUT2 and the ABO gene. So coming to how these chains act. So you can see this is the type 1 chain. This is the type 1 chain you can see. Type 1 chain, let us see what the chains are. So essentially every chain, if you see, they have this R part. Now this R part is a long carbohydrate chain, which is common to both type 1 type 2 chains, right? Now this carbohydrate may be linked to a lipid or a protein, depending upon which it will be a glycolipid or a glycoprotein. So this is the R part remains the same. Now both the chains type 1 and type 2 have N-acetyl glucosamine at sub-terminal portion, and on the terminal there is a galactose sugar, right? Both of them have the same thing, but there is the linkage, the biochemistry linkage which happens between these two sugars is how you differentiate type 1 from a type 2 chain. So you can see type 1 chain there is a beta 1,3 linkage, whereas in a type 2 chain there is a beta 1,4 linkage. So that is how you distinguish type 1 from a type 2 chain, right? Another important thing is I've already told you type 1 chains move freely in the plasma and the secretions. Type 2 chains move in the RBC. RBC are formed on the type 2 chain, right? And they have a beta 1,4 linkage. Now, which enzyme acts on type 1 and which enzyme acts on type 2? So remember, it is opposite. So on type 1 chain will act FUT2 gene, and on type 2 chain FUT1 will act, or the fucosyl transferase, or the H gene. So that is why the H gene, which is the most important gene here, is acting on the type 2 chain, right? That is the FUT1. The FUT1 gene will act on type 2 and the FUT2 will act on type 1 chain. There is another gene called Lewis gene which will also act on type 1 chain. Let us forget that. I'll talk about this later. Now coming to the H activity, very important. Till now we had seen this is the chain, the carbohydrate part along with the N-acetyl glucosamine and a galactose. Now what happens is any individual generally has, all of us, if you see, we have H gene with us. Most of us will have either a homozygous or a heterozygous dose of H gene, capital H. So almost 100% of us will have either one capital H or both capital H. Whenever we have this capital H gene, it will code for an enzyme, like that is the central dogma. So which enzyme will it code for? It will code for a FUT1 gene. So this fucosyl transferase, what it does is it will add a sugar. Like I told you, a fucose sugar will be added to the type 2 chain. So FUT1 gene will act on the type 2 chain and there will be a sugar or a fucose sugar will be added. So this type 1 chain is now changed into an H antigen. So very simply put, whenever you have an H gene will produce an enzyme called FUT1 gene. The FUT1 gene will act on this only on this type 2 chain and it will add a sugar there, which is the fucose sugar. This is the sugar that it has added, and it will change those precursor substance or the type 2 chain into a H antigen. Now we have the H antigen. Now H antigen is a precursor for formation of A and B antigen. So what will happen is, now this is the second gene. So we remember that H gene, if it is present as capital H, then it will produce an enzyme called FUT1. Now similarly, all of us have another gene set of gene which is known as secretor gene, SE gene, or the S capital SE. So even if you have one copy of capital SE, you are going to produce another enzyme which is known as FUT2 or fucosyl transferase 2. It will do the same thing, but it will act on the type 1 chain. Now, where are the type 1 chains present? They are present freely in the plasma and the secretions. So, in the secretion, FUT2 gene will act on the type 1 chain and it will do the same thing. It will add a fucose here. So, once the fucose is added, this also changes into an H substance, but this H substance is present in the secretions, not in the red cells. So, you have H gene which causes formation of H antigen in the RBCs, and you have a secretor gene or the FUT2 gene which will cause presence of H antigen in the secretions. So you have H antigen now present both in secretions and on red cells. Now this is a perfect ground for formation of A and B antigen. So what happens is, so where is this A, B and H substance can be detected? Which secretions when I was talking about secretions? So it is present in almost all secretions except cerebrospinal fluid. So it is present in saliva, tears, urine, digestive juices, bile, milk, amniotic fluid, and other pleural peritoneal fluid and everything. So coming to the ABO synthesis, we have got this H antigen now. This is a type. Which chain is this? This is a type 2 chain because it is a beta 1,4 linkage, and you can see there is a fucose attached. That means this is an H antigen which is present. Now whenever there is a blood group A person, the blood group A person will either be a capital A or a capital A with an O. So that is the phenotype of a blood group A. Also, that blood group A will have an A transferase. This is the third enzyme that I was talking about. A blood group B will have a B transferase, and blood group O will have no transferase. Right? So when an A transferase acts on the existing H antigen, what it will do is it will add another sugar here, which is the N-acetyl galactosamine. So this is a A group, but it will only act if H antigen is already formed. If there is no H antigen, even if you have A transferase, it is not going to add this terminal N-acetyl galactosamine. Similarly, in B antigen, if you see, you have a B transferase. So what it does is on this H antigen, it will add a galactose sugar. But it will add that galactose sugar only and only if you have an H antigen present. If there is no H antigen and only a B transferase is present, you cannot form a B group. Similarly, for blood group O, you can see that there is no transferase, so no sugar will be added. It is similar to the H antigen, and that is why maximum H antigen is present on the O group, because it is not converted into either A or B, right? So this is the basics principle for ABO synthesis. Simply put, in this flow chart, you can see that there is a type 2 chain. So type 2 chains means they are attached to RBCs. On this type 2 chains, if you have even one capital H, the fucosyl transferase enzyme will act on it and it will produce an H antigen. Now this H antigen, once it is formed and you have an A transferase, which is N-acetyl galactosamine transferase, bigger name for that, it will add that sugar which N-acetyl galactosamine and form an A antigen. Similarly, if you have a B transferase, it will form a B antigen, and if there is A and B gene, A and B genes both are present, it will form an AB antigen. If no is present, like in O, it is similar to the H antigen. So what are the genes name? I have simply put it as FUT1, FUT2, FUT3, but the exact name, if you see, the H antigen is alpha 2L fucosyl transferase. And what is the immunodominant sugar? It adds a fucose. So once the fucose is added, the antigen becomes H. Similarly, I told you that for A, it is A transferase. the exact name of which is alpha 3 N-acetyl galactosylamine transferase. And what it's going to add immunodominant sugar is N-acetyl D-galactosamine, forming a A antigen. Similarly, for B, the enzyme name is alpha 3 D-galactosyl transferase, and it is going to add a D-galactose, and the antigen would be a B antigen. So coming to the amount of H, I already specified that in O group, nothing is changing. The H antigen remains as it is. That is why the maximum H is present, or the H antigen is present in O. And A2 versus A1, if you see in the subgroups of A2 has minimal quantity of the antigenic sites, and that is why less conversion, and that is why you can see that H antigen is more in A2 compared to A1. So in the order, in the decreasing order, it is O, followed by A2, followed by B, followed by A2B, followed by A1, and least is present in the A1B group, right? So what is the Karl-Landsteiner law? This we have studied in our high school also, and right from our beginning we have been studying that if you have a specific antigen is present in an RBC of an individual, the corresponding antibody must be absent from the plasma, or vice versa. If the antigen is absent in the RBC, then the corresponding antibody must be present in the plasma. So very simply put, so if you are an A group, obviously you have the B antibody. Similarly, you have a B group, you have A antibody. AB group, you have AB antigen, no antibody. And if you are O group, you have anti-A, anti-B, and another antibody which is anti-AB. The interesting thing about O is that although there is only one molecules of H that is shown, you have H everywhere, but in O the H is maximum because H is not being converted into A or B. So maximum H is present in the O antigen. So A subgroups, ABO subgroup, it was discovered by Van Dujan in 1911. We have one A1 and A2. My colleague is also going to talk about this. The immunodominant sugar in A1 and A2 is, we have already covered it, is N-acetyl D-galactosylamine. So what are the differences between A1 and A2? You can see the number of antigenic site. If you see between A1 and A2, it decreased. There is difference in the precursor of oligosaccharide chain. The amount of transferase enzyme is less, and the amount of branching is less in compared to A2 from A1. So that is how we differentiate A2 from A1. So if you see on the bench work, if we see, we start comparing, you can see that A1, how do we know that this is A1? It is because it will react with both anti-A and anti-A1. So that if you see for a grouping, when you do that, you can see that it reacts with both anti-A and anti-A1. But A2 group is going to react with anti-A, but it will not react with anti-A1. So that is how we discover that this is A1 or A2, and this is the subgroup. Of course, in A2 you can have both anti-B antibody, and many a times you will have in 8%, 1 to 8% of cases, you will have an A2 with an anti-A1. So you might find this antibody, and this may cause a blood group discrepancy at times. So you need to be wary of the same, and you need to know that A2 subgroup can form an antibody which can be anti-A1. And if you see the number of antigenic sites in A1, it is 810 to 1170, whereas in A2, the antigenic sites are quite less, which is around 240 to 290. So these are other subgroups of A. I mean, there are so many subgroups that have been discovered and which can be done, and some of them can be detected serologically. You have AX, you have AN, you have AY, AL, AM. So depending upon whether you have a what type of reaction, you can follow this table. This table is there for your reference, and you can detect. Sometimes you even have to go for a molecular test and not just serologically. You can detect. Similarly, we have a B phenotype. B phenotype, obviously, we have subgroups of B, but they are not as many as A subgroups. B subgroups have been detected quite in a lesser quantity or number, but they are there, and you can see them serologically as well. Very important is Bombay phenotype, or now it was discovered in Bombay. We all know that, but which is now Mumbai. This is a very important concept, but I think more or less you would have understood by now what Bombay phenotype is. It is we all know that we have 100, almost 100%, we have at least one capital H with us. But say those one in 10,000 or 1 in 1 lakh, that individual has both the capital H missing, and it is small h. It is small h, small h. So that person will not be able to form an H antigen. So if there is no H antigen, even if you have A transferase or B transferase, you will not be able to form an A group or a B group. Essentially, it will look like an O group. But how is it different from O group? Is when you reacted with other O group, you will see that it is incompatible. Why? Because in O group, they have H antigen which is maximum, and in this Bombay group you have anti-A, anti-B and anti-H. So you have anti-H reacting with the O group which has maximum H antigen. So you will see that there is florid agglutination, and you can see that this is a Bombay group. So simply stating, this is an H antigen. We all know now this is an H antigen. Fucose is added, and this is the individual who has at least one capital H and forming a FUT1 enzyme, and thereby adding after an A transferase A and B genes. Now suppose you have a non-functional H, that is small h or Bombay group O small h. What happens is then this fucose is not added. Once this fucose is not added, obviously the A transferase and the B transferase cannot act to form an A or B group. And these individuals, since they do not have H antigen also, they form an anti-H. So they have anti-B, anti-B, anti-A and a anti-H. So testing these O small h, that is the phenotype we determined. So when you test it with anti-A1, there is no reaction. That means with anti-B also there is no reaction. But when you add an H-lactin or a Ulex europaeus, which normally would react the O cells which have got this H antigen, there will be no reaction. That is how you discover a Bombay group, or you test for a Bombay group. So this is the reaction that we see in a Bombay group. You would see that anti-A, there is no reaction. There is anti-B, there is no reaction when you start with an A1 cell, B1 cell, it is it. But incidentally on O cells also it will come positive, because you have O cells which have got H antigen and you have an anti-H antibody. This is the Bombay phenotype. Coming to a little more, I mean, sometimes we get bogged down with this topic, is the Para-Bombay phenotype. It is somewhat different from Bombay phenotype. Only thing is, if you remember those genes, you will remember what a Para-Bombay phenotype is. So we had a gene called H gene which was acting on the red cells, and we had a secretor gene which was acting on the secretions, right, on the type 2, type 1 chains. Now, suppose you have H deficient secretor. That means your FUT1 gene is absent, but your secretor gene or the FUT2 gene is present. So what you will have is, you will have red cells which are devoid of that H antigen, but your secretions will have the H antigen. So what happens is, they are not just present on the secretions, they are even present on the plasma, and they keep interacting. Sometimes what happens is, from that secretions or from the plasma, this H antigen gets absorbed onto the red cells. So once that happens, so sometimes when you do the reaction and you start testing for these blood groups, you will see some weak reaction. So that is a Para-Bombay from H deficient secretors. The other type of Para-Bombay can be when your H itself is weak and you are non-secretor. So your H antigen or the FUT1 and gene is very deficiently produced. So that time also you will see those weak reaction, but it is similar to the Bombay group. Only thing is, here your H antigen is weak, there your H antigen was properly present, right? So this is Bombay group. Now coming to the last bit, how Lewis antigen is related to the ABO group system. We are all aware of this. This is a basic native chain type. Which one is this? Type 1 chain, because it is a beta 1,3 linkage, right? Type 1 is chain is there. What happens is, we will have an H antigen, so, and secretor antigen. So generally we have seen that the FUT2 enzyme, that is the secretor enzyme, will act on the type 1 chain. So here the secretor enzyme is going to act. Even if you have one secretor enzyme, capital SC is present, either both capital SC or one secretor enzyme is present, what is it going to do? It is going to attach this fucose chain on the terminal galactose. So this is a simple H antigen that is formed. Now you have another gene which is acting on it, and that is the FUT3 gene or the Lewis gene. What will it do? This will also act on this this chain, and what it does is it is going to add another sugar on the subterminal galactose. And this condition where two fucose are added is known as the Lewis B antigen. When two fucose sugars are added on the type 1 chain. So this leads to the formation of what is called as a Lewis antigen. But sometimes you may have a small se. That means your secretor is absent. So obviously when secretor is absent, you cannot attach the fucose chain, or your H antigen will not be formed. But your Lewis this thing FUT3 gene is still present. So it will attach its sugar. So this is known as Lewis A system. So when both the fucose are added, one from the H antigen and one from the Lewis gene, then it is known as Lewis B. And similarly, if only the secretor is absent, so you cannot form an H antigen in the secretion, but your Lewis gene is active, then you form the Lewis A antigen. So that's how the Lewis system is closely linked with the ABO system, because this also involves working on the type 1 chains which are present on the secretion. So the pearl of wisdom here is, if you are a non-secretor, that means you have small se, you will always have a Lewis A antigen. You will never have Lewis B, because you don't have the H gene which is going to form the H antigen, and that is why this fucose is not added, and only a fucose can be added on the subterminal N-acetyl glucosamine, which is a function of a Lewis gene and not the H gene. So I hope I'm clear in this, and this is how we will go ahead. And thank you and for listening, and have a nice day. If there are any queries or anything I need to address, you can write on the comment and you can tell us.